everybody always thinks computers with technology, but scientific technology is also out there, and you run into it. I see a nurse in the audience. There's a lot of technology in every doctor's office. But also LCC, we have a lot of really great technology that we put into the hands of our students. And this device, I saw about 6 years into being a chemist. The students that I work with see it the first week, and they do some, pretty advanced analysis with it right away. And it's the same technology. It's just the color of the instruments changed slightly over the years. So in a way, and also on the left-hand side of that box there, it's gotten a little more detailed. Over the last several years, students have been, originally the idea was I was going to set up the instrument and they would get the data. And the students kind of wanted to do the development of the process. So they've been doing authentic science, putting together new techniques, problem solving, all of that. And we're going to see kind of some of the physical problems that you have to deal with. Honey is not easy to work with. It's sticky, and our lab balances don't like being sticky. But also there's a lot of details in the computer analysis side that the students have been working on. And quite frankly, I can do a lot of technology, but things like photocopiers don't work for me. And for some reason, the software that runs this is more like a photocopier than a spreadsheet. So students have been actually able to do a lot of great work with it. So this year, the students decided they also, in addition to doing authentic research, and they're registering for a credit every quarter and then spending way more time than that doing the project. In addition to working together and researching with the internet into scientific corners of the internet, they wanted to also get practice presenting. So here we are. We found a way to wedge ourselves into the program for community conversations. And I have to say the students have been preparing everything that's up here. I've been around helping, but the work is authentically theirs. I may guide them. I'm not like having them show up while I'm doing the work. I'm sitting around. I wish I could be drinking coffee, but it's a lab. And supporting them as they're doing great things. So how am I doing on my 5 minutes? They wanted no less than 5 minutes. To introduce the two students that we have with us, Keita Patterson. She's been a student of mine since, I think, 2020. I'm sorry, they've been a student of mine. And so they got to take chemistry during the pandemic over Zoom, but now a lot of lab time is happening. And I don't see the scores, but I'm confident that they just aced their lab final for the year. Also, Newman has been with us for, is this your third quarter? Or second quarter? You joined us in winter. And Newman immediately started making a difference with the lab work, which was usually we're like, hey, in about a quarter, you'll start to feel, what's going on. Newman is able to really just jump right in there, has some very direct interest in the types of technology, and it relates to his career plans. So I'm going to turn it over to you guys. And thank you guys for coming out. And hopefully we'll get to say at the end of the day that we taught a Jeopardy champion some new material.
Thank you, Armando, for that wonderful introduction. So I'd like to start off with some of the goals of our project. And some of the goals of our primary goal of the project is to detect small amounts of pesticides in honey up to the part per billion range. Currently, we're able to detect pesticides in honey samples, but we aren't able to detect specific pesticides at very small quantities in the honey. And eventually, we, as mentioned earlier, we look to expanding our reach into a community outreach project where we'll be accepting honey samples from farmers or local beekeepers in order to test their samples to make sure that they're within acceptable level of pesticides. We won't be any actual accreditation, so it won't actually affect them, but it's always nice to know those types of things. Unfortunately, this slide was, we had an outage. We had 3 students here that were going to present, and the extra student was in charge of some of these bits. So we're working it in here today. We're trying our best.
Currently at our state where we are at this moment in lab, we have the tools available to be able to identify pesticides, and we are working in lab protocols, including getting intake samples from honey farmers and trying to just piece the process together. Currently, we have the ability to detect presence of about 8 to 10 different pesticides to 0.4 parts per million in dummy samples. We are far from meeting our goal, but we are slowly but surely making progress towards that goal.
So one of the first things you may be wondering, what is a pesticide? I'm sure everybody's heard the word pesticide before, but you may not be familiar with what exactly qualifies as a pesticide. Here I broke down some of the various pesticides. Here's a list from the California Department of Pesticides. Unfortunately, Washington State, we don't have our own governmental body to test for pesticides. California is the state that has the most funded and well-developed body for testing pesticides, and they classify pesticides as any of these types of chemicals, from insecticides, herbicides, rodenticides, repellents, attractants, fungicides, disinfectants. The list just goes on. Basically anything that interacts with a pest in order to get rid of it. And attractants are on there, usually baits, known as an attractant to get an animal into the trap. You may be asking, what's a pest? A pest is just any living thing, such as a rodent or a weed in a garden that you don't want to be there, that's disturbing the productivity of whatever environment it's a part of.
And a huge thing about pesticides is it's this common idea that pesticides are getting rid of a problem and it's a solution. But as we've seen historically, that's not the case. And in fact, we're doing more detrimental damage to our environment than just getting rid of a pest.
So pesticides, they're really important because they impact, or they're really important to the environment because they're used in farming to allow us to have food on our plates. But also pollinators are very important to the environment to allow us to have food on our plates. Pollinators are actually responsible, some places say up to 1/3 of every bite of food is responsible for a pollinator, especially bees included. And unfortunately, pesticides, especially neonicotinoids, directly impact the ability for pollinators such as bees to survive and directly have an impact on their reproductive health. Here's a few examples of how pesticides are added to the agricultural environment. They start off, systemic pesticides are pesticides applied to seeds. Those are used in order for fungus, small organisms and fungus can affect seeds and prevent them from germinating. So pesticides are then added at the beginning of the plant's life, and that can, especially neonicotinoids, will stay within the plant throughout its entire growth cycle. Later on, spray application is applied to the plants as well in vegetation, especially with insecticides to prevent the fruiting bodies of the plant from getting disrupted by the pests. This is usually the interaction where we see within honeybees, where bees will interact with the fruiting bodies of the plants, especially flowers, where they'll take in pesticides at that point, especially the pesticides covering the pollen, and then bring it back to their hives. You can see in this diagram, it's essentially that the spray application, bees going to their flowers, leaving their flowers, and at that point, they're absorbing the pesticides into their bodies, and then they bring it back to their hive. And that's the most detrimental part of it, where they encountered the most pesticides, because by bringing it into their hives, they're adding pesticides not only to the honey, but the honeycomb of the hive. So all organisms within that hive are then affected by the pesticides. And this isn't a problem that just affects bees. As mentioned, like the seeds, even land invertebrates and birds are going to be impacted, because if they eat the seeds, they're going to be affected by these pesticides. And the reason why we chose, or the reason why it's so useful to use bees as a way of finding these pesticides, is bees are very good at going to plant, especially flowering plants, and they can take up little bits of pesticides. Then by analyzing their hive structure, both the honeycomb and the honey, you're able to get an average about how many pesticides are in that area just collected by those bees. Here's a study I found showing the neonicotine rates of bees in a cornfield. The yellow graph is bees in the cornfield and their effect, versus the blue graph is the unexposed bees. They took, I believe, a wild sample of bees, but this chart shows that in a realistic field experiment where you're taking or you're sampling a colony of bees and watching their lifespan in a actual agricultural environment, you can see there's a significant impact on the lifespan of the bees. It's not, the pesticides don't completely kill the bees, they just drastically reduce their lifespan. The first chart here is the time of their last flight, which is the last time they were alive, essentially, because it's very hard to track a bee. You can't just put a little tracker on each of the bees and follow them around. You have to figure out, okay, this one flew their last time on this day. And it's just very shocking to see. That's a matter of, I believe, weeks almost, or a full week between the top of one of the control group and the top of the treated group. These bees aren't getting to the crops as long, and this can lead, especially in late flowering crops, if the bees aren't out in the fields actually pollinating the plants at that point, it can lead to collapse of those species of plants, and for the late flowering crops at least.
So pesticides are clearly not only a problem for the bees themselves, but also for our ecosystems that rely on pollinators to be able to reproduce. Then there's also a whole other aspect of, we as humans ingest a lot of honey, and we ingest plants and other crops that these bees are going to. And so by being able to measure pesticides in honey, we are being able to see how much microparticles of toxic waste we are consuming as humans, and also the effects that it has on the ecosystem that is not just honeybees.
I found it interesting that over time, especially in the more recent years, our use of pesticides has reduced. Here's a brief history of pesticides. Humans actually have been using substances as pesticides dating back to almost the start of history. But in more recent years, starting in the 1867, Paris green was added to plants, specifically in France. It's a mixture of arsenic and mercury, I believe. But two compounds many people know are very toxic today. However, back then they thought using Paris green on their beans was a more effective solution to preventing the American weevil. It was American beetle of some sort that became an invasive species in 1867 and was totally decimating their fields. Well, after introducing Paris green, they found the effects of the arsenic and everything was actually very detrimental to the people consuming the plants, and thus started the history of pesticide testing after that point. But even from the beginning of synthetic pesticides, we ran into problems. The next significant pesticide I'm sure everybody's heard of is DDT, also known as Agent Orange. Or I may be mixing those two up actually. But DDT, such a horrible chemical that it was banned in the US in 1972, almost or a full 33 years after it was initially synthesized and used in the USA. After DDT was banned, the USA decided that, hey, we don't know enough about pesticides to keep just applying them blanketly to all the crops in the area. What can we do in order to reduce the amount of pesticides and dangerous chemicals people are consuming? So in the 80s, they introduced the OECD, which is I had it written down notes, but it's essentially a collective of rules that are a generalized standard for people to apply throughout the US specifically. Unfortunately, that's a really important factor to note. Rules are only good for the country that is using them. And something like pesticides is a global issue. So borders don't matter in terms of pesticides. Just because Canada or Mexico may have stricter or more reduced pesticide laws, the bees and the plants aren't going to care about the pesticides that are 20 miles away across the border. They're still going to be impacted, maybe less so because there is some distance, but overall, the standards set in America are only for dealing with in local communities. And even as we saw earlier, California has much stricter standards on what pesticides are allowed to be used and how much can be used, and even when they can be used at certain times of the day compared to even our state of Washington, which is incredibly detrimental because our crops are actually some of the most impacted by pesticides and neonicotinoids because Washington State's primary crop is apples. And apples are pollinated by honeybees, and honeybees, unfortunately, get sprayed with neonicotinoids. And in California, they actually primarily use almond, or their primary pollinators are of almond trees, which are a more domesticated bee. The European honeybee, which is less affected by certain pollinators as the native pollinators in Washington State. So that's also a big variation between species. Are the wild native species affected by the pesticides, or are the domestic species being bred to be pesticide resistant? And just because the pesticide resistant strains we get in our domesticated European honeybees aren't as affected by the pesticides doesn't mean that the native species of bees aren't going to die off because of a similar amount of pesticides introduced to them. Here's a description of the Bee Project currently. A little background on the Bee Project as a whole. Here are some of our past members. Dr. Armando Herbelin, you may know, is one of the few continuing people in the project. The project goals continue to be the same. They said they wanted to calibrate methods for common pesticides. We have completed that goal. We can mark that off. We have calibrated the method for common pesticides. However, we have moved on to pursuing specific pesticides and documenting down to a part per billion count on the amount of pesticides in the sample.
And I think it's even better to note that this project was a brainchild of students. This wasn't a proposal that Armando came to these initial students. You see Brian, David, and Caitlin. This was something that was student driven. a goal of making a difference in our community. And then they formed this idea of let's test pesticides in honey samples. But how do we do that? And each year, even though it seems like it's been a while since 2018, we've overcome things like COVID. And through these trying times, we've continued to build our testing methods and get one step closer to achieving our ultimate goal of being able to be an accessible resource and be able to test quantities for specific pesticides in the parts per billion.
This is one of our past members, Sarah. She did groundbreaking work within the project. Some of the internal standard we're currently using was actually synthesized by her. We're waiting until the fall. We're not waiting. Until the fall, we have a set amount of internal standards we're going to be able to use because she had such amazing technology she learned from the OChem classes at LCC that she was able to bring into the Bee Project. Over here, we have our method for sample collection. We have a jig, as we call it, that Alex made using his 3D printer at home, a little vial that we store samples in, and then a small syringe that we actually extract 1 gram of honey out of honey jar and put it into there. We found that that's the most effective way of dispersing honey into a sample, and we'll go further into how we determine that that's the most effective way of dispensing a single gram of honey within, I believe, 5% consistency rating on all the samples.
Yeah, and I'm going to go into a little bit of our methods of how we've gotten our samples prepped. So it starts with something we call our internal standard. And this is a chemical that, as mentioned previously, Sarah synthesized, and it's essentially what we use as a measurement against the other pesticides. It's a collection from the Oregon pesticide standards. So we know the chemicals in there and we know the concentration. And that's the goal of the internal standards. From there, we have to be able to mix honey within a small vial without making a mess and create a balance of these two that we can then set into the GCMS. So our main tool that we are using in the Bee Project is called the GCMS, or gas chromatography mass spectrometry. And we're going to play a little fun game where I'm going to rattle off a whole bunch of science things and Newman is going to translate for you. So the idea behind a gas spectral mass spectrometry is that it combines 2 techniques, gas chromatography and mass spectrography. So first, what we do is we put our sample that we've created into the GCMS. During this, where it's going to be the first phase, which is the gas chromatography, where the sample is going to be heated very rapidly so it vaporizes and is injected into a long coiled tube known as the chromatographic column. The column is typically coated with a stationary phase, which interacts differently with each compound injected into it. Inside the column, the compounds are separated by their affinities to the stationary phase. Essentially, as compounds interact more strongly with the stationary phase, it takes longer for them to travel through the tube.
So essentially, we take a sample and we extract a little bit of that sample up, and then it goes into this oven. And that oven heats up the sample in the tube, and then depending on how much the sample weighs, depends on where it is in this tube. And then that tube shoots out a particle into an ionization source.
Yeah, so as each compound exits the GC column, it enters the mass spectrometer. Here, it is bombarded with electrons, causing it to fragment into smaller charged particles known as ions. Then the ions are produced in the ion source are accelerated into the mass analyzer, which separates them based on their mass to charge ratio, or M to Z. The separation allows for identification of different chemicals based on their unique spectra. The separation of Ions are detected by a detector, also known as electron multiplier, which generates an electrical signal proportional to the abundance of ions at each NZ value. What we get is a mass spectra unique fingerprint. Each compound gets its own identification or peak in our spectra data.
Once it goes through the ionization source, it's then thrown into the mass spectrometer, essentially, where it's all analyzed using a lot of sophisticated programs, and we're just given these peaks that we're able to use other really sophisticated programs to analyze. And looks, after all that analyzation, you get this, which still needs to be analyzed a third time. But
So this is a graph of essentially, as we talked about, how these compounds exit and then they're charged with ions. They're given an energy of sorts. And from there, it bases them on how they arrive. So their energy levels and their mass levels. It really focuses on comparing samples. And so we get these peaks, and then as our job as students and scientists is to go through and be able to individually label things. We often clean it up so we get these more simplified peaks with data numbers.
each of these peaks is basically a correlation of each of the chemicals we're looking for, except for some of those peaks don't mean anything. Some of them mean there's something we don't want in there, we're detecting something we don't want, and then some of them we just don't know. So it's a guessing game until we add it to another software where we are able to compare it in a library and be like, ooh, this really tall peak, that's probably pretty accurate. Did somebody else get this really tall peak here? And we're like, hell yeah, we did it right, we got it, one. And
it's also important to note when we talk about this graph, we're talking about our time, which is our bottom, versus our abundance. So this is essentially retention time, how much time it spends in the machine and how much comes out of it. From there, we go into our library and we start identifying individual compounds. And for the most part, this is something that our computer can do. However, we did run into the problem where a lot of pesticides are complex compounds that we have to individually put into our library, meaning we have to test that, look at the graph it creates, and then input that into our own library. And this just is a simple example of how we do that comparison. So this top graph is a reading that we got from one of our labs, and the bottom graph is a pesticide chemical that we were testing. And so we have to try to line up these peaks and try to identify the chemicals based on how close they are. And here's another great example of how we stretch that graph out so we can compare it. And as you can see, our timing lines up. So we have our 120 lining up with 120 and a peak in a similar place. So it's a lot of comparing, and sometimes it feels like guesswork when you don't quite know what you're looking for, but it really comes down to being able to identify things and know what you're looking for.
so the next steps of the Bee project is going to be specifically looking for each of the pesticides within the sample. And as it sits, we don't have it down to an exact number. We're working with a 0.4 parts per million, which is still pretty small. 0.4 parts per million could be argued to be 400 parts per billion, but we would still like to get that number into the double-digit parts per billion range.
Especially, I feel like it's important because there are so many pesticides that have such a big effect at little concentrations. So being able to identify at a further parts per million would be more beneficial for our research. So now we're going to kind of just briefly talk about the sample prep and like how we created this, along with some of the issues that you might have heard Armando talk about earlier, is our sample prep. On the basic idea, we take a little bit of honey, 1 gram to be exact, and inject it into a tube. From there, we use our internal standard and we add that based on weight. And we make sure that we're trying to be consistent through each sample, because if we mess up one sample, then we don't have accurate data to compare. Then we add deionized water to create a uniform volume among all samples. And so this is a little video, and I'm going to pause really quick, of how the direct immersion solid phase works. As you see, we have our vial and it's being pierced. Then small fibers being injected into the tube, and then it's going to attract the fiber and then place the vial back. And that fiber is what's going to be tested, is what's on that small piece.
So here are some of the trials and tribulations we experienced throughout the project. And as mentioned previously, preparing the samples took a lot of time. And the biggest problem with that, the number one problem we had to occur starting at the beginning of this year, was actually dealing with the viscosity of honey and reducing it to a point where we could actually work with the honey and put it in exact, or as close to 1 gram as we could get without going over or under, which on paper, I mean, I'm sure everybody could think of like, oh, you just take a spoonful out, you can get pretty close, but
But another thing to keep in mind is we couldn't get any honey on the sides of the vial without compromising our machine and equipment. And so we had to find a way how to get a decent amount of honey into a really small hole without getting anything sticky. And so after many, many brainstorming sessions, we came up with this convoluted solution that involved one of our members, Alex, creating what we call an injection jig from his 3D printer that allowed us to be able to insert a syringe or pipette into the vial without spilling honey. And so this is just a little brief example of one of our first versions of our injection jig and the process that we did in order to fulfill that process.
I cut the pipette to the 1 mil line right here. I cut the bulb off of the end like this, just so that we get the flat part available. And then I cut slits into the sides. So that we can shim it on, which is a bit of a difficult process. That should, the pipette is set to 1 mil.
So as you see, that black thing is our injection jig. So that was our very first prototype that we created, and it was a bit of a process. And then we had the issue of we're working on this project to benefit the environment and to benefit our health, while we're also producing a lot of plastic waste by cutting up stuff in order to just be able to make our sample preps. So we wanted to figure out a way how we could get rid of wasting half a pipette, which led us to our second.
Well, on top of that, our consistency wasn't quite there. Go back to the slide. So the other thing we were intending to do is to heat up the honey, and that resulted in this amazing contraption made by our wonderful Rachel Sparks, who are, in order to heat up the honey, you have to hold the honey in the water while boiling the water, while testing the heat of the water, and also having a thermal plate. So it's all these wires and everything. So the poor student who is the newest at the project would have to set this device up at the beginning of every day at the start of my quarter. So I was the lucky student who got to set this thing up when I first joined. Luckily, we were able to get past this stage, so that's no longer a task for the lucky newbie of the group. But
then there was an aspect of human error, as you noticed that we were actually using micropipettes in order to inject it into this. And an issue is that sometimes we goof up. And in order to prevent these goof ups that could be detrimental to our testing, we had to frequently undergo what we called our pipette validation experiment, which is essentially where you take a little pipette, fill it with water, and get a test tube and just measure how much you are piping at each time for about 20 to 30 trials over and over again until you get a consistent result. And so it's just little things like this that made it hard to be able to get the consistency that we are looking for. And it really came to having the tools available and having the time available. So this was the original injection jig, where we had a pipette, a plastic pipette that we would cut up in order to fit over top of one of our micro pipettes, and we would place it through this 3D printed jig and place it on top. From there, we would be able to put the honey in without causing any spillage.
the original jig was a two-part system. You may have noticed our second jig became one part. This entire process was continually worked on. After the micropipettes, we moved on to the syringe, as I noted earlier, where we were able to perfectly fit. Actually, the jig was made first. We discovered the syringes much later on. So our second jig, made by Alex as well, was one piece of 3D printed material that you were supposed to be able to get one of these micropipettes to fit through perfectly. Doesn't always work that way, unfortunately, but conveniently enough for our applications, that jig worked perfectly for the little syringe you found. And we found that when you suck up about 0.95 milliliters of honey into that syringe, you're able to distribute about a gram consistently, because you don't run into the air bubbles you may have noticed in the video when you're distributing honey from a micropipette.
So now that we've talked about what our Bee project does, I want to talk about the further implications of this project. First, I want to look at it from a student perspective. As Armando talked about, we have access to tools that not a lot of science students or STEM students get to use. We get to build a deeper understanding of how we can use our laboratory tools to better understand the world. We are actively pursuing real-life applications, things that are not just in school specific. We're looking for further things that we can do to make an impact on the community, but also better our understanding, better our skills as a student. As Oxford University wrote in January 2023, students who have access to high technology and equipment beyond those at their skill level grow to meet the challenge. If we didn't know how to use the GCMS, Armando sat us down, walked us through one time and said, now you do it yourself. And it's moments like those where it's the idea of you learn by doing, you learn by growing, and opportunities like this having new technology to grow our skills makes us better students. The next part is the implications for our community. Hopefully through this project, we are going to get better regulation for bees, safer honey consumption, reproductive for bees and pollinators as a whole, and a better understanding of how pesticides affect us, while also creating an accessible resource within beekeeping communities. I want to talk about how just in the last couple of years, we have seen a huge increase in regulation for bees, and this is through the continued awareness. And I believe that if we can do our part by showing the parts per million that we see in pesticides, we can better regulations. This is compared between 1960 to today, and it talks about application rates versus nowadays and how over the last couple of years, we've seen a decrease in applications. We've seen better regulations and we've seen more research done. We're moving towards a better future just by bringing the awareness. And even if we haven't accomplished a lot in this project so far, we have made huge strides to building a community-focused idea of bettering honey, bettering consumption, and bettering the bee community. The FDA wrote that companies continually improve the efficiency and safety of their products to meet high regulatory standards and give farmers the tool to produce safe, abundant, and affordable foods. As we continue our work within the Bee Project and we make movement towards having a better understanding of pesticides and how pollinators are affected by that, we're going to follow this idea of safe, abundant, affordable food. I also want to do a little comparison of how our test compares to other tests. According to the Washington Department of Agriculture, the average testing protocol is about $158, and that requires a lot of bees, tons of honey, and shipping of all of these goods. That is a lot for a small business. This is a lot for honey and beekeepers who are just getting into the business. But if we were able to fulfill this project, we would be able to make a community resource where the only thing that they would have to provide is a very small amount of honey and the shipping to get it to us. We would be bettering our community by growing resources, while also creating a safer environment for bees and humans to coexist.
In conclusion, our overall goals of pesticide identification, the original goal of the 2018 project has been achieved. We are able to identify whether or not the presence of pesticides in honey does exist. And I mean, we knew there would be pesticides in there, but we are able to determine, yes, this specific sample of honey has one of, I believe we're up to 8 pesticides specifically we're able to detect for at this point. We were able to handle, especially the honey, as previously noted, getting down to 1 gram of honey in each of the samples exact or within 5% was a significant hurdle for us to overcome. So getting through that has been an enormous achievement. And finally, the valuable lab experience. Even if this project hasn't achieved a community outreach quite yet, all the students within the project have gained practical lab experience that they have used further on, especially someone like Sarah, who I wish we could have today, but she's busy off in her 4-year university studying and doing this chemistry skills that she's learned during this project.
Our further goals, as we mentioned earlier, is to maintain our progress and further our knowledge base. We want the Bee Project to be a community outreach. We want to have this as an opportunity to make an impact on our communities. And we want to expand our database and be able to identify more pesticides quicker. And that concludes our presentation. We thank you guys for all giving your time and attention to us. We really appreciate it. If you're interested in learning more about the Bee Project, you can scan that QR code. That it will actually send you to Armando's email. So
It-
Is there another way to get
yeah, we are available on campus often. It's really hard because we are
I mean, online.
we are currently working on building our website, but it's been kind of hard with our shifting of students.
This QR code will take you to a little bit, fair, but it should take you to more information about the project. That's what that is assembled.
And there's also the email address for Dr. Herblin down there as well.
We may be able to get a postable link for it. We'll look more into that in the future. But we would also like to note that this is a project based on a volunteer basis. So if you know anybody who may be interested, any students at LCC in the future that would like to join our projects, we would greatly appreciate any students that you can send our way.
I have a question to start us off here. Are there other community colleges, four-year universities, or other communities that are doing projects like this in other areas of the nation?
I believe it's hard to say because this specific project is something unique to LCC. And my hope is that there are other community colleges or universities that are providing this research opportunity. But I also know that the idea of students being able to have access to these high quality without, teachers holding your hand through the process is kind of a revolutionary idea.
Well, I've got a question and then I've got something to say. My question is, how many pesticides are there? Do you even know? Are there so many that you can't keep track of them?
So I'm sure there's an exact number somewhere out there, but that falls into what's your definition of pesticide? Like you saw California has a different definition to the state of Washington. Are we saying a bait's a pesticide? Are we saying, but a rough number, hundreds of thousands out there.
Ooh.
Yeah, and we have to also take into account 99% of those pesticides probably have little to no effect on us. They are specifically there, or they're usually biological control mechanisms used in order to specifically target certain pests. Like if you have a bacteria strain, it may specifically attack certain fungus on things and have little to no effect on any of the other things. And that's just an issue of the classification.
And that is one of the challenges that we've been working to overcome is our process for choosing the pesticides is looking at the most common pesticides used and then including our area and then ones that directly affect bees. And this is like a compilation of a list that we have to put together ourselves. And it's hard because there are resources online and we do look at the Oregon pesticide standards, but it's hard because it's not something that has a lot of attention, which is why our group thought it was so important to do a project focused around that.
Actually, go to the next slide. I think we have a list of all the next one. Yeah, super small, sorry. I wish we could have enlarged it, but there's not a ton on here. Here, let's see if we can blow it up real quick for you. This is actually a list of all the pesticides we can test for in the OPS. So there's only 40 something we're actually testing for that we think have or are documented in the OPS, but the actual number of pesticides in total is much greater than this list alone.
I've got something to say because I bought a bag of apples once and I ate one. I could taste the pesticide on it. I really had to scrub them and I still couldn't get that taste off. They gave me a free bag of apples when I told them about it, but that doesn't, I could taste it. I don't know how they probably don't even clean them, do they? When they pick them.
I'm not sure I wouldn't imagine they're spraying apples themselves. Maybe the neonicotinoids could stay in them. Um, that's very strange. I didn't look into the actual consumer products, pesticides, like how you're spraying them on the plants. I was more concerned with the effects on the pollinators themselves, but yeah, that I wouldn't be surprised if there are some pesticides that end up on the fruiting bodies, like the neonicotinoids. I I doubt it would be a toxic pesticide. We do have some standards in Washington that keep a toxic pesticide from being sprayed, like I mentioned, the Paris green. We do not allow arsenic or anything on the apples, but it could be something that caused you discomfort. It wasn't a good taste. It's probably, and that's not pleasant, and we don't need that. But it probably prevented a bug from being on there too. So it's like, which would you rather have?
Did you find much variation in viscosity?
In the honey itself, we were only using one type of honey, but we did find old honey is much more viscous than new honey. So as soon as we got a fresh sample, it started working a lot better for us.
That was a challenge that we had to overcome. Is general consensus is honey, when warmed up, does become easier to work with. But once you warm it up, it generally starts crystallizing once it cools back down. And so then we have this bottle of honey that is now crystallized and much harder to work with. So we kind of went through this trial and error of, first we're like, what if we tried to freeze the honey? And then we found out that honey doesn't really freeze well. Then we moved on to, okay, maybe we can just kind of like work with it the way it is. Also didn't seem to work. And finally, we just started heating up and getting it to a certain viscosity that was easier to work with.
And at some point, are you thinking of considering different varieties of honey, like clover honey, to see if there's differences there?
Oh yeah, that is a big goal of ours, is to be able to have a broader intake of different types of honey. We've been looking at getting honey samples from like California residents and seeing how pesticides are different geographically.
Well, thank you so much. This has been a wonderful take, not just through the Bee Project, but also what the sciences do here at LCC and also the scientific method in general. I know I learned a lot. So thank you all for joining us, and thank you to the students of the LCC Bee Project for sharing their information here. Thank you all.